The medial temporal lobe has attracted the attention of neurologists, neurosurgeons, and psychiatrists alike. And before we cut further into the temporal lobe, we will emphasize some of its important features. The medial temporal lobe contains two of the most celebrated limbic lobe structures, the hippocampus and the laterobasal cortical amygdala, which are hidden from view in the depth of the parahippocampal gyrus. In our last coronal section, we encountered the most rostral part of the amygdala. As we continue further back, we will reach the hippocampus, and according to this drawing, the two structures will overlap for quite a distance. The parahippocampal gyrus is characterized by a hook-shaped structure, the uncus of the parahippocampal gyrus, which is tucked away underneath the mesencephalon. The entorhinal cortex, which covers a large part of the exposed surface of the parahippocampal gyrus, is an important gateway to the hippocampus formation and the amygdala, which have been exposed by opening the lateral ventricle and scraping away part of the parahippocampal gyrus, as shown in the hemisphere to the right. The amygdala has been artificially separated from the hippocampus to enhance the image. In reality, they are contiguous. The arrows on the left indicate how impulses converge from the entorhinal cortex via a cascade of association pathways from higher-order sensory and multimodal regions in different parts of the cerebral cortex. The entorhinal area can sometimes be identified by the naked eye because of the presence of small bumps called verrucae. It looks like chicken skin or orange peel. Occasionally, individual bumps are as beautifully displayed as in this photograph taken by Gary Van Heusen. The verrucae correspond to islands of cells in the superficial cell layer of the entorhinal cortex. These cells provide most of the fibers to the perforant path, colored blue in this drawing, which carries the multisensory information to the hippocampus formation for the formation of long-term memories. The perforant path perforates the subiculum on its way to the hippocampus, thus the term perforant path. Hippocampal projections back to the entorhinal area and other parts of the cortical mantle, which are important for memory storage, are colored red in this drawing. Fornix fibers to subcortical regions, many of which originate in the subiculum, assemble in alveus before they exit through the fornix. Here are some pictures from Gary Van Heusen's collection showing Alzheimer's changes in the entorhinal hippocampal system. To the left, a Nisser-stained coronal section with the general location of the perforant path projecting to the hippocampus and the dentate gyrus. To the right, a matching section with 88 immunostaining, which shows early stages of neurofibrillar rectangle formation within the cell islands in the entorhinal area and in their projection fields in the hippocampus and dentate gyrus. In advanced stages of Alzheimer's disease, the entorhinal area is inundated with senile plaques and neurofibrillar rectangles, and hippocampus is effectively disconnected from the entorhinal area, of which there is little left. At this stage, however, most of the cerebral cortex is affected, the various association areas relatively more so than the primary sensory motor areas, as reflected here in the paracentral lobule on the medial surface of the hemisphere.